I'm John Strum, and this is Real Talk MS. It's April 15th, and we have a lot to talk about. But before we get into today's episode, I want to send along congratulations to Dr. Stephen Hauser and Dr. Alberto Escario, who were both named winners of a Breakthrough Prize, an award that's been nicknamed the Oscars of Science. Dr. Hauser and Dr. Escario were each honored for their groundbreaking research in multiple sclerosis. Dr. Hauser's 4 decades of cutting-edge research as a physician-scientist has revolutionized our understanding of the genetics, immunology, and treatment of MS, and led to the development of B-cell therapy, which has proven itself to be highly effective in treating all forms of MS. I'll add that Dr. Hauser is one of the nicest people you'll ever meet. And if you'd like to get to know Dr. Hauser, I'll invite you to listen to my conversation with him in episode 310 of this podcast, and you'll find that link in today's show notes. Dr. Escario observed that MS mostly affects people in the Northern Hemisphere, making it much more rare in tropical countries and areas close to the equator. This led him to wonder whether a virus could be involved. After almost 20 years of research, Dr. Escario and his team were able to confirm a link between MS and the Epstein-Barr virus. And while the Epstein-Barr virus is not the cause of multiple sclerosis, it has been identified as a necessary condition. Both Dr. Hauser and Dr. Escario have made game-changing discoveries that continue to lead researchers to new answers and new information as they continue to unravel the riddle of multiple sclerosis. So once again, I'll say thank you and congratulations to both of you for this very well-deserved honor. While we're talking about well-deserved honors, last week at the American Academy of Neurology's annual meeting, Dr. Amit Bar-Or was honored by the National MS Society and the American Academy of Neurology as this year's winner of the John Dystel Prize for Multiple Sclerosis Research. Dr. Bar-Or is being honored for multiple achievements that have advanced our knowledge of neuroimmunology, precision medicine, and biomarkers in MS. At the American Academy of Neurology's annual meeting, I had an opportunity to ask Dr. Bar-Or how some of the latest discoveries in MS research were going to impact people with MS. But before we get to my conversation with Dr. Bar-Or, there are a few other things that you should know about. Last fall, reporting from ECTRIMS, the largest MS research conference in the world, we talked about the results of the phase 3 clinical trials for Tolebrutinib, an investigational disease-modifying therapy from Sanofi. If you'd like to learn why so many experts are excited about this potential medication, I'll invite you to check out my interviews with Dr. Robert Fox, the principal investigator of the phase 3 clinical trial that evaluated Tolebrutinib for non-relapsing secondary progressive MS, and Dr. Jiwan Oh, the principal investigator of the phase 3 clinical trial that evaluated Tolebrutinib for relapsing remitting MS. And you'll find links to those interviews in both audio and video in today's show notes. As I shared with you a couple of weeks ago, Tolebrutinib is currently making its way through the approval process at the FDA, and just this past week, the results of those phase 3 clinical trials were published in the New England Journal of Medicine. If you'd like to review the details of these clinical trials, you'll find those links in today's show notes. Anyone living with MS can tell you that one of the things they tend to hate about an MRI exam is how long it takes. And that has everything to do with the number of different scans that are required in order to accurately account for the number of lesions and determine the amount of brain shrinkage that a patient may have. This isn't only inconvenient for patients. It also makes it more difficult for researchers to run clinical trials that are designed to track the effects of a particular treatment on lesions. The good news is that a research team at University College London has developed a new artificial intelligence tool called MindGlide that's capable of accurately calculating lesion load and brain atrophy in someone living with MS from a single MRI scan, regardless of the type of scan. The initial goal of this project was to develop a tool that would analyze the hundreds of thousands of existing MRI scans in hospital archives to provide researchers with a better understanding of MS and how MS treatment affects the brain. The research team trained their AI by having it analyze 4,000 MRI scans from 3,000 people living with MS. These scans were acquired using almost 600 different MRI machines. Applying its algorithms to these scans, MindGlide was able to identify patterns, and through that pattern identification, it learned how to identify lesions. Once MindGlide was trained, it was tested on nearly 15,000 MRI scans from 1,000 people with MS. And the result was that MindGlide was better at identifying MS lesions than state-of-the-art computer tools, and its results agreed with the experts whose job is to review MRI scans. MindGlide also proved to be effective at determining the effects of a treatment by analyzing clinical trial data. In one test, the research team compared MS lesion load and brain shrinkage in people with primary progressive MS who were treated either with Ocrevus or a placebo. The team did this comparison using MRI scans that were not even taken for that purpose. The results of the analysis showed that the trial participants who had received Ocrevus had lower increases in lesion volume and slower rates of brain shrinkage compared to the trial participants who had received a placebo. The correlation between lesion load and disability levels was greater using MindGlide than with standard of care AI tools. In other words, MindGlide outperformed the AI tools that are available today to analyze MRI scans. Interestingly, the team analyzed different MRI scans to compare lesion rates among the participants of a clinical trial that compared simvastatin against a placebo for people with secondary progressive MS. The results showed no difference in lesion volume changes over time. However, the analysis revealed significant slowing in brain shrinkage among the trial participants who were given simvastatin. Initially, we'll see MindGlide being put to use in research, but the team that developed this tool sees it being further developed for use in the clinic, when a single MRI scan will help neurologists better understand a patient's condition. Now, if you'd like to review the study that describes the development and validating tests for MindGlide, you'll find a link in today's show notes. When it comes to MS disease-modifying therapies, can there be too much of a good thing? In the case of Ocrevus, newly published evidence from a phase 3 clinical trial indicates the answer is yes. Roche, the manufacturer of Ocrevus, conducted a clinical trial to determine whether higher doses of Ocrevus showed additional benefit in slowing progression among people with relapsing remitting MS. And the results of that trial show there was no additional benefit gained. Prescribed and approved dose of Ocrevus is 600 milligrams every 6 months. And in this clinical trial, participants were given doses of 1,200 milligrams and 1,800 milligrams, depending upon their weight. Results show that the rate of disability progression was low and consistent with rates of disability progression that were observed in previous studies that used the 600 milligram dose. So the results of this study can be filed under nothing ventured and nothing gained. On the plus side, Roche has also shared additional data from their clinical trial of a 10-minute subcutaneous injection of Ocrevus. A full year of data from this trial shows that the subcutaneous injection demonstrated a near-complete suppression of relapse activity and a minimal increase in lesion load among people with relapsing remitting and progressive forms of MS. This is consistent with outcomes from the traditional IV infusion of Ocrevus. So in this case, while there may not be improvement in the outcome, because the outcome is already pretty outstanding, there is a benefit in the broad accessibility and convenience of a 10-minute subcutaneous injection compared to a 2 or 3.5 hour IV infusion. Biotech company FibroBiologics has announced that their investigational cell therapy, called CYMS-101, has succeeded in increasing myelin production and myelin repair in not one, but 2 mouse models of MS. When we talk about cell therapy on this podcast, we're usually talking about stem cells. But not this time. This therapy is made up of connective tissue cells called fibroblasts. Fibroblasts are one of the most abundant cells in the human body. They're involved in wound healing, and they're one of the 2 types of cells that can regenerate bodily tissues. That other cell type is stem cells. FibroBiologics explains that compared to stem cells, larger numbers of fibroblasts can be obtained more easily. The cells themselves are more robust, and they require less care than stem cells, making it cheaper to manufacture and maintain a fibroblast therapy. FibroBiologics has already completed a phase 1 clinical trial to determine the safety of CYMS-101 in humans. Trial results showed that CYMS-101 was safe and even showed early signs of efficacy. Currently, FibroBiologics is looking for funding or a partner to come in and support launching a phase 2 clinical trial. Given the attention being paid to remyelination, coupled with the initial success of CYMS-101, let's hope the company finds that willing partner. Meanwhile, if you'd like to learn more about CYMS-101, you'll find a link to a detailed report on the FibroBiologics website in today's show notes. It seems like we've been discussing several research breakthroughs in this episode of the podcast, and discussing game-changing research is the ideal segue to my conversation with this year's winner of the John Dystel Prize in Multiple Sclerosis Research, Dr. Amit Bar-Or. Dr. Bar-Or's work has driven meaningful advancements in virtually every important area of MS research. And in a moment, you'll meet my guest, Dr. Amit Bar-Or. Dr. Amit Bar-Or holds the Melissa and Paul Anderson President's Distinguished Chair at the University of Pennsylvania, where he serves as Chief of the Division of MS and Related Disorders, Director of the Center for Neuroinflammation and Experimental Therapeutics, and member of the Leadership Council of the Colton Centers for Autoimmunity. And Dr. Bar-Or is this year's winner of the John Dystel Prize for Multiple Sclerosis Research. First, congratulations, and tell me, what does this award mean to you?
Well, thank you, John. It means a great deal. It's a real honor to get the Dystel Award. It's 30 years now, since 1995. The late Don Patey was the first awardee of the Dystel Award. And I think, there's nothing like peer recognition, and we all work hard, we all do it passionately, and really to be recognized by colleagues, it means we're at least on the right track.
A few months ago, I heard you discussing how being diagnosed with what you referred to as the new MS represents a very different patient experience compared to, say, someone who was diagnosed several years ago with the old MS. Can you talk about that?
Sure. I think the concept of the new MS is really to reassure people as they're facing a new diagnosis, most of whom may have known somebody, have a neighbor, have a relative who's had MS for years. And we are now hopefully able to catch people earlier and earlier in the course of their MS and have a much greater impact on limiting injury from MS, so that the hope is that people who have MS will live with MS as though they don't have MS. That to me is the new MS, the prospect that I think is not an overpromise. I mean, we always want to be realistic as well, but the realities have changed sufficiently. And while there's still very important unmet needs, at least for the relapsing aspect of MS, we should now be able to stop people from having both the relapses that they experience and also those that happen under the surface and can contribute to damage over time.
There's a drug candidate currently making its way through the regulatory approval process. And this particular therapy represents a new category of disease-modifying therapies called BTK inhibitors. You've investigated the role of BTK inhibitors. Can you explain what makes the category of DMTs different and potentially important?
So the Bruton tyrosine kinase inhibitors, or BTK inhibitors, are a class of drugs that target a molecule called BTK that's present in a variety of cells, but in the immune system, particularly in B cells and what we call myeloid cells. And the myeloid cells include also not just the circulating or peripheral myeloid cells, but tissue myeloid cells, including the microglia cells in the brain. And we think that B cells and myeloid cells, on one hand, may be contributing in part to the relapsing disease, but very importantly, within the central nervous system, they may represent very important targets of progressive MS, so that the B cells that seem to traffic and then compartmentalize within the CNS and the microglia are known to be abnormally activated in the context of MS, thought to contribute to progressive MS, and having brain-penetrant BTK inhibitors, some of which are more penetrant than others, but having a brain-penetrant BTK inhibitor has raised the prospect of being able to target within the central nervous system some of the underlying biology of the progressive MS.
Another area of MS research that you've contributed to is the identification of potential biomarkers for MS. Can you remind our listeners about what a biomarker is and then explain the benefit of biomarkers both in clinical trials and clinical practice?
Sure. So a biomarker really is a biological measure that has some clinical meaning. Now that, of course, is the general definition, but there are many different types of biomarkers. So if I can measure something in somebody today that predicts how he or she will do in the future, that's a prognostic biomarker. If I can measure something that tells me this person has condition A versus B, that would be a diagnostic biomarker. If I can measure something that informs me about the state of activity of a person's MS, for instance, more active, less active, in ways that we can't see on the surface, that is a biomarker of MS activity. So in general, biomarkers are tools for us to be able to capture something meaningful about a person's experience with a condition that is not readily visible in some other fashion, on clinical grounds, by imaging and so on. Now, imaging can be considered a biomarker as well, and we use imaging in MS for a long time, but I think the biomarkers that you're referring to are probably more those that relate to fluid biomarkers, where we can measure, for instance, injury products of the central nervous system in the blood. So it allows us within the blood to get a sense of what might be the consequence of injury within the central nervous system, a molecule such as neurofilament light chain. Another one, glial fibrillary acidic protein, or GFAP, is associated typically with astrocytes within the central nervous system. And when it is elevated also in the blood, it seems to inform us perhaps more about the non-relapsing progressive aspect of MS. Now, these are relatively early days in trying to understand how to use them in the clinic, because of course for a biomarker to be useful, it needs to be measuring what you think it's measuring, it needs to be measurable in an accessible sample. Some things can be measured in spinal fluid but not in blood, so it's not that they're not useful, but you're not likely to be doing lumbar punctures again and again to monitor people over time. And it needs to be done in a way of measurement that is reliable and affordable and accessible. And all of those are features that make a biomarker either a good one or not a very good one. We are beginning to get a handle in the MS world about biomarkers that may distinguish the relapsing biology from the non-relapsing progressive biology. And we've come to realize that much of those biologies are happening under the surface. The person living with MS may not be aware of them, and we don't yet have ways of measuring them in terms of what we see clinically. But if we could measure under the surface the presence and the relative presence of the relapsing biology versus the progressive biology, we will have a much better sense of the state of that individual biologically and a much better sense of which type of treatment we might want to discuss, especially now that we think treatments that are going to be targeting not just the relapsing aspect, but also the progressive aspect.
When it comes to rethinking the paradigm of patient care, you and your team have developed something called the Precision Neuroimmunology Platform, and I'm hoping you can explain what that is and how it could improve patient outcomes.
So we are not the only ones who are interested, of course, in this concept of precision medicine, which aims to try to more specifically characterize an individual's state of MS. We, in our platform, try to integrate multiple different domains of measurement that include clinical domains, things that we can see and measure in terms of the person's experience with his or her MS, imaging measurements, both the ones that we obtain routinely clinically, but also additional research ones, and then additional biological measurements that could be done in the blood and or in the spinal fluid. And then collectively, this provides us with a certain sense of this individual's overall profile. And we hope that this is going to give us a sense of how to capture the relative contribution of the different biologies to the individual's MS and guide treatment at a more individualized or more precise level.
So putting a treatment plan together that really becomes individualized based on the specific aspects of that specific patient in front of you, as opposed to the generalized, this is what we do for relapsing remitting MS, for instance.
In essence, yes. I mean, we learn through evidence-based medicine, which we all subscribe to, and that comes from, in part, clinical trials. But the clinical trial will tell you this medicine, on average, decreases MS activity by 90%. There are people who do better than the average. There are people who don't do as well as the average. And it's not a one-size-fits-all. The term individualized medicine, or personalized medicine, was used previously, but then there was also the realization that if you have 1,000 people, you don't have 1,000 treatments, right? So the idea of precision is to be more precise than a one-size-fits-all, but it's not quite individualized.
Your work has driven meaningful advancements in virtually every important aspect of MS research. As you look at the research landscape today, what excites you?
Well, John, I think we are very much stepping over the cusp, I think, of targeting non-relapsing progressive MS, finally, although partially. I don't think this is the last cry, but I think if you look back 30-some years ago when the first interferon was approved for MS and limited new relapsing disease activity by about 30%, similar to what we've seen now with the BTK inhibitor on progressive MS, that was partial effect, but it was the first time that MS had transitioned from a non-treatable condition to one that was treatable, and yet we knew we could do better. So this, I think, is igniting the field, and we'll have a lot more focus on this very tough nut to crack, which is non-relapsing progressive MS. Of course, we need to figure out how to fix things that are already injured. So the repair and regeneration field is something that we're beginning to get traction on, and I very much hope that within a few years we'll have some breakthroughs in that arena as well.
Well, Dr. Amit Bar-Or, congratulations once again on being named the winner of the 2025 John Dystel Prize for Multiple Sclerosis Research. And thanks so much for talking with me today.
Thank you, John. Always a pleasure.
That's going to wrap up this episode of Real Talk MS. Real Talk MS is powered by the National MS Society. And you can share this episode of the podcast by letting your friends or family members know that all they have to do is point their web browser at realtalkms.com/398. You'll find that link in today's show notes, so you can easily copy and paste it right into an email or a text. And if you have a minute, I hope you'll visit the Apple App Store or the Google Play Store and download the free Real Talk MS app for your iOS or Android smartphone or tablet. It's the best way for us to stay connected. The app will automatically download the latest episode of Real Talk MS. You'll be able to access any of our past episodes. You'll be able to save your favorite episodes. And it's a great way for me to share bonus content with you. The app is free, so I hope you'll download it today. I'm John Strum. Thanks for listening. Stay safe and make healthy choices.